Thank you so much for the wonderful introduction. Yes, I can hear you. Can you see my slide?
Yes, we can. You can proceed.
Thank you. Today, I am going to present to you about one of the studies that we have been doing. There are many studies and many analyses that we've been doing, but today I chose just to come and show you about the microbiome, the levels of microbiomes, and how this can be linked to the increase in risk of non-communicable diseases. So as the title goes, changes in the gut microbiome and virome link urbanization of South Africans to increase in risk of non-communicable diseases. Non-communicable diseases are a diverse group of diseases that include obesity, diabetes, atherosclerosis, and some cancers of the digestive tract. They present a primary health threat in high-income countries, but are now increasing in developing countries. In South Africa alone, about 60% of deaths in 2017 were attributed to the non-communicable diseases. There is a widely supported hypothesis that changes in diet and lifestyle associated with urbanization as part of the economic development in sub-Saharan Africa are the main drivers of NCD risk. However, the underlying biological mechanisms are not fully understood. So when we eat food, food is digested in the digestive tract by enzymes, and the soluble nutrients are then absorbed into the body. Any undigested remaining foods, it ends up in the large intestine where it is metabolized by resident gut microbiome. What is intriguing about this gut microbiome is that it has a large amount of genetic material, and it also has a higher metabolic capacity than that of the liver.
Yes,
So as a result
it's
of
better
that
now.
capability of high metabolic capacity, remains of different kinds of foods, can be metabolized by the gut microbiota into a diverse range of products. Here, where you see in the blue arrow, we are pointing at short chain fatty acids. These are products that are produced when remains of fiber are fermented by the gut microbiota. And here in red arrows, you then see uh, products that are produced, metabolic products that are produced when some of these foods like meat and maybe fatty foods is also metabolized by the gut microbiota. The reason why we have blue arrows is because short chain fatty acids such as butyrate, have been found to be protective against the development of colon cancer and have been associated with good health benefits. And in red arrows here, we have various nitrogenous compounds, and maybe sulfur-containing compounds, as well as the secondary bile acids. And these have been found to be associated with dysbiosis. Dysbiosis simply means imbalance of the microbiome, like when it's not healthy, as well as inflammation and disease. So we then hypothesize that the transition to high-fat diets, it promotes then the dysbiosis of the microbiome, reflected by the suppression of these butyrate-producing bacteria, and then the increase in levels of bacteria that will produce the secondary bile acids, as well as the distinct patterns of bacteriophages or the viruses that are associated with these bacteria. And we aimed at investigating how the ongoing transition from traditional to urban lifestyle affects the gut microbiota and its metabolome by by making comparisons in the fecal microbiome and virome of the rural and urban Amaxhosa people. So our study, which is a pilot study, is a cross-sectional observational study that is based in Cape Town, a city that for many years has been receiving migrant populations from within the country as well as outside the country. Those that came from within the countries, they originated from the province called Eastern Cape Province. And as a result, in this province, we set up a rural cohort for our study. And also in the urban area in Cape Town, in a township called Khayelitsha, we set up an urban cohort. And in both studies, we then recruited a total of about 45 healthy, middle-aged men and women. So from these participants, dietary information was collected using food frequency and 24-hour recall questionnaires, and it was analyzed by linking to food composition tables from the South African Medical Research Council. From these participants, different kinds of samples were collected. We have fecal samples, urine samples, saliva samples, as well as food and other environmental samples. Now, the fecal samples were analyzed by sequencing the bacteria and the viruses. And the resultant sequencing data was then characterized for composition, meaning what is there, diversity, what are the different kinds, and then abundance, meaning the level. So before I tell you about these bacteria and the viruses, I just want to take a detour and tell you what we also found out in the other preceding studies where we really made comparisons of the amounts of nutrients. So what we see here is that for many macronutrients, the levels were higher in the urban than in the rural areas. Now, I don't know, there may have been talk about fiber, because fiber is so interesting. We talked about the short-chain fatty acids being the fermentation products from the fiber. And, you would expect that with urbanization, because we are talking about the increase in risk of non-communicable diseases, maybe the fiber is decreasing. But we found that fiber is also increasing as we are, people are moving from rural to urban. And furthermore, when we make comparison, I also made a summary of comparisons here, and I showed here that we have shown that the dietary fat is tremendously increasing from rural to urban. And associated with that, we see that the sum of the bile acids that come from these fats, we see that there is a higher increase in the urban than rural. However, the significant microbial gene that is involved in converting these fats into secondary bile acids, namely this one called BICD. With BICD, we see that there is no difference between urban and rural. And we talked about the butyrate earlier. Of course, with butyrates, we did not see any significant differences between rural and urban. However, still remember that the fiber consumption is higher in urban than in rural. So now coming back then to the bacteria. What is interesting about the bacteria is that there was significantly higher level of alpha diversity in rural than in urban. Now with alpha diversity here, as I show species richness, there are different number or different kinds of species present. And also the figure on beta diversity, there was a significant difference in beta diversity. Here it just shows that there's all the samples that were collected in the rural and urban, they all cluster in distinct groups, showing that the composition of the microbia of the microbiome is very distinct and different. And with differentially the abundance of the microbiota or of the bacteria. Here I am showing all the red bars that shows the genera, the bacterial genera that were abundant in the urban area, and the blue bars, all those that were abundant in the rural area. And the red arrows that you see there, they are pointing to the bacteria that in many studies have been found to be associated with colon cancer and bowel inflammation. Here we see Fusobacterium. If you read many papers about colon cancer, you'll find that Fusobacterium is always implicated. If you read many papers about bowel inflammation, you'll find that Bilophila is always implicated. Now, coming to the blue arrows, we are pointing at the genera that in many studies have been associated with healthy rural lifestyle. Now, the bacterium, Faecalibacterium and Prevotella, these are known short-chain fatty acids producers, or they are known degraders of carbohydrates. And of course, this is very concerning. You know, if you see the bacteria that we can call bad guys are higher in urban community, and the bacteria that you can call good guys are higher in rural community. This is really concerning, because this is what we think might, be linked then to the increasing risk of the non-communicable diseases. Let me share also about the viruses. About the viruses, there was no difference in the alpha diversity in the viruses between rural and urban. However, when it comes to the clusters of samples, we see that maybe samples from one origin, like from the rural with the blue dots, they tend to maybe cluster together, although not very definite. And when we look at what is differentially abundant, here what we see are that there are some viruses that will attack the bacteria, or that will be associated with the bacteria in the rural as well as in the urban areas. And it was surprising to see that some of these bacteria, like Bacteroides, and everywhere in the rural and urban, it is susceptible then to this viral infection. And also, we also noted that Faecalibacterium here, this Faecalibacterium, remember in the other place, it had a blue arrow. And that blue arrow shows that it is abundant in the urban area. However, when we look at the data here, we see that it is being susceptible then to infection of its own viruses in the urban area. And we think that this might be a way to even control the levels of this Faecalibacterium, because the level was lower in the urban than in the rural. However, some of these data that I have presented here, they still need to be further analyzed. So we need to even extract more information, because there is something that is interesting there. And in summary, from these studies and the few that I have presented today, the gut microbiome environment, they change in composition from rural to urban. However, the bacterial, not viral diversity changes to urban. The differential abundance of the bacteria that promote tumors and the decreasing of those bacteria that produces the short-chain fatty acids in the urban cohort is very concerning. That was the figure with the blue arrows and the red arrows. The role of gut bacteriophages, or the viruses as we call them, and their interaction with bacteria and the human host require further investigation. And in conclusion, taken together, all these results and those presented in other meetings, they provide evidence that changes in the dietary pattern and the gut microbiota and the virome with urbanization may be predisposing to the development of colon cancer and other non-communicable diseases. Further studies are required to determine whether the restoration of specific components of the traditional diet will stop the accelerating rise in non-communicable diseases in South Africa. And with that, I wish to thank you and to acknowledge all these institutions that have been instrumental in making this research a success. Thank you.
Thank you, Dr. Matsepo. That was a very good presentation that now brings in the macrophages, the viruses that also are associated with the microbiomes and their potential effect. Of interest, as you've indicated, is that difference between the rural and the urbanized community in terms of the distribution of those very significant microbiome of various bacteria the good ones and the bad ones. They tend to be more as you move to the towns and tend to westernized diet than what we have in the rural. But what I would like, maybe clarification, the treponema, is it the treponema or I saw some bacteria there of interest which was of blue color, other than the one you mentioned. I think it was the treponema. Am I right, Dr. Matsepo?
Yes, Prof, you're right.
Yeah. And it caught my attention. I don't know whether it is the Treponema pallidum or the Treponema, which species of Treponema?
Oh, with this analysis, we are able to detect up to the genus level. We cannot detect up to the species level.
Oh,
So
okay.
everything right now is all the different genera.
Okay.
It would be nice, to get,
Yes,
to
to the
further deep
species.
down and find the species or even the strains.
Yeah, I would be interested because Treponema pallidum, this side of the world, is a risky business for us. If it's present, I don't know. But if we could check the species of the Treponema that was identified, it would be very interesting. And maybe another interest is why it was marked blue. That would be also of interest. Is it also a good bacteria as by your study? The treponema.
Treponema. For now, what we can say is that treponema is associated with healthy rural lifestyle.
Okay.
So in general, but
Okay.
I think there may be still problematic species or strains here and there. So it will be interesting to really study this.
Okay. I'll be interested
Can I just say something? Could I add a comment, please?
Yes. Yes, go ahead.
So it's a very good question, but it must be remembered that, as Matsepo is saying there, is that with our sequencing, we isolate different large groups of microbes which contain many subspecies. Treponema pallida is not the main component of the treponema groups that we actually measure.
Okay.
So it's the same in, for instance, for C. difficile, and C. difficile is something that is devastating in hospitalized patients, and patients can actually die when they get an overgrowth of it. But it's a natural component of the microbiota. So if you've got a healthy microbiota, it tends to sort of suppress the pathogens or the overgrowth factor. So there are several other studies from Southern Africa which show that treponema is actually very common amongst rural Africans, and it's associated with health, as Matsepo says. So this makes it very difficult to say these are good bugs and those are bad bugs. If they're living in a balanced community, and the balanced community is kept by a balanced diet, then in fact it prevents pathogens occurring and pathogenic effects.